To observe how endocrine organs communicate, researchers often use controlled animal models. In one such setup, scientists monitored hormonal baselines of a healthy mouse. The experimental variable was the introduction of propylthiouracil, or PTU. This drug specifically targets the thyroid gland, inhibiting the enzyme required to synthesize thyroid hormones and effectively halting production. The expected physiological outcome was a state of induced hypothyroidism, marked by a sharp decline in circulating thyroid hormones. However, the blood chemistry revealed a contradiction. While the thyroid was shut down, TSH levels had skyrocketed, remaining significantly higher than the healthy control group. This creates a paradox. The reacting organ was suppressed, yet the TSH signal surged in direct inverse proportion to the drop in T3 and T4. Solving this puzzle requires us to stop viewing endocrine organs as isolated chemical factories and instead model them as nodes in a closed, electrical-style circuit. Decoding this specific feedback circuit reveals the structural logic of endocrine regulation, transforming complex hormone interactions into a system of predictable first-principles responses. The nervous system relies on high-speed electrical impulses for immediate coordination. The endocrine system operates through chemical signal, secreting hormones into the bloodstream to reach distant target tissues. The anatomical bridge between these two systems is the hypothalamus, located at the base of the brain. The pituitary gland sits directly beneath it, acting as the secondary command center for metabolic signaling. The hypothalamus and the anterior pituitary are linked by a highly specialized closed blood vessel network. This hypophyseal portal system ensures a unidirectional flow of hormones from the hypothalamus directly to the anterior pituitary, bypassing the general circulation. This arrangement allows the hypothalamus to use tropic hormones. These chemical messengers do not act on end organs like muscle or bone. They exist purely to command other endocrine glands. This vascular link is specific to the anterior pituitary. The posterior pituitary relies on direct nerve axons for its connection to the brain. The portal architecture allows the brain to function as a centralized command center, using the anterior pituitary as a biological amplifier to broadcast instructions. We can map this command structure through the thyroid axis. The chain begins when the hypothalamus releases thyroid-releasing hormone, or TRH. TRH travels through the portal system to bind with receptors on the anterior pituitary. The anterior pituitary then releases thyroid-stimulating hormone, TSH, into the main systemic bloodstream. TSH circulates until it reaches the thyroid gland in the neck. The thyroid node then uses iodine and tyrosine to synthesize its final products, the hormones T3 and T4. These hormones enter the circulation to set the body's basal metabolic rate. However, T3 and T4 also loop back up to the command centers in the brain. This brings us to the mechanism of negative feedback. The final hormones in the chain physically bind to receptors on the hypothalamus and the anterior pituitary. This binding sends an inhibitory signal that actively suppresses the further release of TRH and TSH. This creates equilibrium. The system produces only enough hormone to meet metabolic needs, preventing a runaway reaction. It functions like a mechanical thermostat. Once the target temperature is reached, the thermostat signals the furnace to shut off. In a healthy endocrine axis, the final product is the very thing that restricts further production. Returning to the initial experiment, the drug PTU blocks the thyroid's ability to assemble its hormones. Because the gland is chemically paralyzed, the production of T3 and T4 ceases. As existing hormones naturally degrade, the circulating levels of T3 and T4 drop to near zero. This means there is no inhibitory signal reaching the brain. The negative feedback loop is broken. The hypothalamus and pituitary register a critical lack of metabolic hormone. In response, the command center triggers a massive surge of TRH and TSH to stimulate the thyroid. But because the thyroid is blocked, it cannot respond, leading to a permanent, unrequited loop of TSH production. The TSH spike seen in the blood tests was the correct mechanical response of a healthy command center trying to fix a broken factory. By analyzing the ratio between a high upstream tropic hormone and a low downstream direct hormone, we can identify the specific point of failure. This provides a diagnostic rule. In a negative feedback circuit, primary glandular failure presents with high upstream tropic hormones and low downstream output. This is the universal architecture of the endocrine system. In the adrenal stress response, CRF from the hypothalamus triggers ACTH from the pituitary, which in turn triggers the release of cortisol. That cortisol then loops back up to inhibit both CRF and ACTH, maintaining the same loop structure we saw in the thyroid. Mapping these circuits allowed medicine to intervene in previously fatal metabolic conditions. By understanding the signal paths, researchers could isolate and replace specific missing hormones, as evidenced by these early insulin treatments. Administering a missing hormone like insulin or thyroxine acts as a substitute for the downstream signal. It restores the physical inhibition at the top of the loop, bringing the system back into balance. Mastering endocrine physiology requires shifting the focus from memorizing individual molecules to mapping signal flows. Once the circuit is defined, the hormonal imbalances caused by organ failure follow a traceable, logical path.